Welcome to this video lecture on contingency tables. Contingency is a word we don't use very much in everyday life. It simply means dependence. Does one thing depend on something else? Another word we can use is association. Is one thing associated with something else? So in contingency tables, or tests of association, or tests of independence, we are exploring to see if one set of categories is in some way connected or dependent on another set of categories. For example, if we use a certain medical intervention, is a patient more likely to recover than if we use a different intervention? Or in an industrial process, if we use a certain version of the process, will we have fewer defective items than if we use some other version of the process? So there are so many applications. In sport, if a certain person takes the kick, is there a better chance of a score than if someone else takes the kick? So we have all these options of exploring how one set of categories is related to another set of categories. I say categories because it's not measurements we're talking about. It's categories. It's whether a certain outcome occurs or not. Or where there are a number of outcomes, whether some of those outcomes are more likely. For example, in the Titanic disaster, we had 4 categories of people on board. First class, second class, third class, also called steerage, and crew. And we can analyze those data to see if the survival of a person was in some way dependent on their category. Were first-class passengers more likely to survive the disaster than third-class passengers? In fact, they were. Okay, so a contingency table takes the 2 sets of categories and puts one set of categories in the rows of a table and the other set of categories in the columns of the table. So the table will have at least 2 rows and 2 columns. For example, a simple medical contingency table explores whether the person received a certain treatment or not in the rows, and whether they recovered or not in the columns. So then in this table, we put in the numbers. We populate the table with the numbers that we have observed by gathering some data. We count how many there are in each cell. How many people receiving this treatment recovered, did not recover? How many people receiving the alternative treatment recovered or did not recover? So we populate the table with the numbers based on our observations of the data. And we call these the observed frequencies. This is what we observed when we looked at the data, when we gathered the data. We then use the data in the margins of the table to calculate the expected frequencies. What numbers would we expect to see in the cells of the table if there was no dependence, if all of the interventions were equally effective? So we also insert into the table the expected frequencies. And then we explore to see, are the observed frequencies better than the expected frequencies? So, for example, based on the null hypothesis that the intervention is no better than any other approach, we would expect to see equal proportions of patients recovering in both the control group and the experimental group. So we compare the observed frequencies with the expected frequencies based on the null hypothesis to see if there's a big difference. And we do that using a statistic called chi-square. And if the chi-square is large, then the p-value is low. The data are telling us it is unlikely that there will be such a big difference between our observations and our expectations unless the null hypothesis is false. So a small p-value, as always, leads us to reject the null hypothesis. So the null hypothesis says there is no association, there is no dependence, there is no contingency. And the alternative says, oh yes, there is. There's an association between these 2 sets of categories. Now we can have a 2x2 table, or we can have a table with any number of rows and any number of columns. And we're still exploring whether one set of categories in the rows is related to another set of categories in the columns. And in order to collect the data for a contingency table, we need to have at least 5 observations in every cell, or to be precise, an expected number of at least 5 in every cell. But the way we gather the data, there are 3 ways to gather the data. We can either take a simple random sample of the entire population and then afterwards classify each observation according to which row and which column it's in. For example, we can take a random sample of all the patients and then inquire which treatment did the patient receive, A or B, and did they recover, yes or no. So a single random sample of all the data, then classified in 2 ways. Or else, we can take a sample from each row. That is, we can take a sample of the patients who received the intervention and a sample of the patients who did not. And in the case of each of those samples, inquire whether each item is classified as recovered or not recovered. So a sample from each row, and then classify each observation according to the column, so it goes into the correct cell. Or the third way is take a sample from each column and classify it according to the row it's in. So take a sample of patients who recovered and classify each one by inquiring, did they receive the experimental or the control treatment? And a sample of patients who did not recover and classify them according to each one. So in certain cases, it would be more sensible to approach the sampling in one way rather than the other. But all 3 methods of sampling are equally valid. We take random samples either way, either one random sample or random samples where there are rows, or else C random samples where there are C columns. We end up with the same outcome. We end up with a table of data in rows and columns, and we have a count in every cell which we use for doing the analysis. You can read more about this in section 5D, Contingency Tables.